So I'm happy to present Valerie. She did her bachelor's at UNAM in New Mexico, not New Mexico, Mexico City, and she did her PhD at UNAM also. She has been in my group for 5 years now, which is crazy. She was a postdoc to start off, and now she became a research associate. And this year she was appointed Provost early career fellow. Yeah. She's going to talk about the lipid divide. One of the biggest questions in the origin of eukaryotes, and one of the biggest questions in biology, which she's trying to solve.
Hello, everyone. I'm absolutely thrilled to be here. And I want to start by thanking Brett and Sean for inviting me. So today is extra special because there's a very important guest in the audience. My mom, she's visiting me. Since I started working at UT, it's the first time that she's seen me present in front of an audience since my PhD defense. So today I'm gonna talk about something that I'm very passionate about, and the lipid divide. So let's start by how we can use the microbial metabolism of modern organisms to understand early life. So throughout my entire scientific career, I have adopted kind of an integrative approach that combines fieldwork, bench work on molecular biology, and computational work to try to understand a big question. What are the extreme limits of life? So basically, we know that microorganisms thrive in have colonized nearly every environment on Earth. And so specifically, my research has been focused on these three environments. Stromatolites from deserts and arid environments for my PhD. And for my postdoc, I study hydrothermal vent systems and hot springs. So these environments hold a very special place in the history of life because they represent the earliest, let me see if I have the pointer. There we go. The earliest evidence of life on Earth. So the geological record indicates that the earliest evidence of life on Earth is from hydrothermal vent systems in Canada or stromatolites from Greenland and Australia. And so one of the other things that we see when we look at the history of life is that life has been mainly microbial. 90% of the history of life on Earth has been microbial. And so once we have this fossil record, paleontologists and geochemists employ different techniques to link specific signatures in the fossil record to specific microbes. They use techniques such as isotopic fractionation or secondary ion mass spec to kind of link specific physiologies with specific microbes. And so despite we have this evidence, there's no physical or chemical evidence from fossils that are preserved enough that we can link a specific or reconstruct in detail the earliest microbes. So, and also we're not always that lucky to have the DNA of a microorganism preserved or microorganisms preserved in the fossil record. As we mentioned, 90% of life in history of life is being microbial, but there's only a few instances where we can have the evidence, the geological evidence for this. So how do we reconstruct the life in the past? So life on Earth has mainly two repositories of information that we can use to reconstruct life in the past. One is the genetic diversity of modern organisms, life on Earth, and we can trace their evolutionary journeys. And the other is the physical evidence that life has left in the past. And this is called biosignatures that are preserved in the geological record. And so what are these biosignatures, right? So imagine we have an ancient environment, such as a hydrothermal vent system, and we have an ancient microbial community. So now let's focus on an ancient microbe, as we see here. This beautiful ancient microbe illustrated by one of my dearest friends, Amilcar. We have an ancient microbe that has their DNA, proteins. When this microbe dies, and all of the organic matter is lost and is degraded in the environment. However, in their membranes, they have these specific molecules that serve to help the membrane for stability, fluidity, etc. One of these molecules, as we know, is cholesterol. And so these molecules also, which is an steroid and hopanoids in bacteria, these molecules, when the organism die, these molecules are preserved for millions of years, even though there's some changes at the level of the hydroxylation sites, but the main structure of the molecule, it's preserved through the fossil record, right? And so then paleontologists such as Jock and Brooks can go into the fossil record, actually extract these lipids, characterize these molecules, and then start asking questions and make interpretation about the organisms that today are synthesizing these specific molecules. And then geobiologists such as Paula Verlander can ask questions about which genes synthesize these molecules, which organisms today are the ones who are producing these molecules, what are the ecological niches, etc., and follow the evolutionary trajectories of these microbes. However, in the fossil record, there are also molecules that we don't know who's producing these specific lipids. They're called orphan biomarkers because we don't know the specific organism that it's producing them. So when Jochen came to Brett and us, he was asking, do you guys know if archaea today synthesize these specific molecules? So that's when the journey began. And so we team up across different disciplines, paleontology, geobiology, genomics, computational biology, and microbial ecology. And so by leveraging the power of each of these disciplines and the strength of each disciplines, I suggest to call this micropaleomics, right? And so this micropaleomics, the idea is basically understand whether organisms today can synthesize the molecules that are in the fossil record. Why are we interested in archaea in the first place? So let's just go back a little bit and remember that life on Earth is divided in 3 domains. Prokaryotes, which are single-cell microorganisms that have their DNA dispersed in their cytoplasm with no nucleus. And so these prokaryotes, I am calling prokaryote bacteria and archaea. However, eukaryotes, eukaryotes, they encompass different organisms such as plants, fungi, us, right? And so the defining feature of eukaryotes is that the DNA is enclosed in this membrane with a specific and defined nuclei. So it was after the genomic era that it was very clear that eukaryotes or eukaryotic cells were some sort of like a chimera. They had a specific characteristics that were very clear and associated with bacteria, such as the fatty acids in their membrane and the way that they obtain energy in carbon. But also eukaryotes have specific characteristics that are archaea-like, such as replication, transcription, and translation. And so it was thought that eukaryotes had specific genes that were called eukaryotic signature proteins, because back then it was very clear that those were unique to eukaryotes. But it was in 2016 that everything changed with the discovery of Lokiarchaea. And so here we see the North Atlantic Ocean and between Norway and Greenland. And this is a hydrothermal vent system that is called the Loki's Castle. And so in this place, it was obtained different genomes from something that the scientists haven't seen before. And they call it Lokiarchaea because of the place that was found. And so these genomes were obtained via a method called metagenomics. And so what's metagenomics? Metagenomics basically is obtaining from any given environmental sample, hydrothermal vent system, hot spring, soil, anything you can imagine. You get all the DNA, all the organisms that live there, obtain their DNA, extract their DNA, send the DNA to sequence, get millions and millions or billions of pieces of that DNA, which are called reads. And then you start to assemble the puzzle to see which piece of DNA belongs to each organism. And this is a very computationally heavy method that basically you have a puzzle and you have to kind of figure it out which DNA belongs to which organism. And the process of doing this is by putting together the pieces of DNA into larger contiguous pieces of DNA, which are called contexts. And based on different methods, you can group these contexts into clusters and start defining which specific genomes. And these genomes are actually called metagenome assembled genomes, or MAGs. So basically, with this method, what allows you is to reconstruct genomes from the environment without having to culture them. And why this is important? Because we can culture most of microbial life, especially from extreme environments. We're talking about extreme conditions that we can replicate in the lab. We have extreme temperature, extreme pressure, conditions that are very hard. So metagenomics was really a game changer in microbiology because of this. It allows us to reconstruct genomes from the environment. And with this, we can start analyzing their evolutionary trajectories, the potential role in the environment without having to culture them. So when we started asking these questions, in 2016, when the scientists started asking these questions about Lokiarchaea, when you put them in the tree of life, something pops up, and it's that eukaryotes, meaning us and all of our complex life forms, branch within Loki, right? So this was very interesting. And then years later, different scientists from all over the world, including Brett's group, started discovering genomes from other members of Asgard archaea. And this started the Norse god mythology of naming these genomes or naming these groups as the Norse god mythologies: Loki, Heimdall, Thor, etc. And what was really interesting about these organisms is that when you look at the genomic potential, you see signature proteins that were thought to be unique to eukaryotes. You have genes that code for cell division, trafficking machinery, everything that was thought to be unique of eukaryotes, these organisms have. So it was really interesting to start to kind of understand the genomic potential. And so to start, what does this all mean, right? So back in the days, in the 60s, 70s, there was a theory. There's a lot of evidence. There's a lot of biological evidence that suggests that complex life, eukaryotes, form via endosymbiosis, which means an archaeal host engulfs a free-living bacteria, an alpha proteobacteria. That bacteria later became the mitochondria. And so this gave rise to eukaryotes. So when Asgard archaea was found, it was very clear that the ancestors of Asgard archaea were the ones that were involved in this event, right? So, but we're talking about 2016, 2018, and there was a lot of skepticism. A lot of people were like, oh, this is contamination, like this cannot be true. And so it was not until 2020 that a group of scientists from Japan actually in cold culture, the first representative of Asgard archaea, was the cover of Nature. It was 3 years ago. And all of the hypotheses, all of the genomic-based hypotheses were confirmed by having this culture, right? And so after this important breakthrough in 2020, Asgard-Archaea research has been expanded exponentially. So our understanding of Asgard-Archaea has evolved from having a few representatives to having a large amount of different members of Asgard-Archaea. So in light of this new diversity, in light of all these novel groups, there's one question. Are Asgard-Archaea still the closest ancestors, or are still branching within eukaryotes? And which of these groups is the one that's specifically closest to eukaryotes? So in order to address this question, and to tackle this question in a monumental effort that took 5-6 years, we were able to position the location where eukaryotes fall in the tree of life. And so now you see here eukaryotes. Now we were able to predict that Hodarchaealis is now the closest ancestor to eukaryotes. So now that we were able to identify the closest ancestor that potentially gave rise to eukaryotic life, the next question was, okay, so we have our lovely hot ancestor, how this hot ancestor was involved in the eukaryogenesis event, how this ancient Hodarchaea ancestor contribute to the emergence of life. We don't have fossil record of hot, we don't have anything that can take us back to how this started, but we have a method that we use to kind of trace back the potential metabolism and physiologies of these organisms. So imagine that we have these hot ancestors that live in the past, and that via different evolutionary trajectories, selection, speciation, different processes, it gave rise to the modern day Asgard-Archaea that we are seeing today. So now imagine that also this hot ancestor had in their DNA, their genetic potential, specific genes, and those genes follow different evolutionary trajectories that might not be the same as the ones that the organism goes through, because genes can be changed between organisms, and that can kind of mess up the phylogeny. So the genes not necessarily follow the same trajectory as the organism. And so one way in which we do this is to reconcile the gene tree, the species, and the gene tree. And so by doing this, the main goal of this process is that we infer the probability that any given gene family was present in the common ancestor. You have certain probability. And so by studying the ancestors of each of the archaea through the phylogenetic tree, we can see whether that gene was lost, whether that gene was transferred, was duplicated, etc. And so by reconciliating all of the gene families present in the current Asgard-Archaea with the species tree that we now know, we infer the potential metabolism of the Asgard ancestors. So I want you to look at this specific cell, and this is what we think, based on this gene tree reconciliation method, that the Asgard ancestor that gave rise to all of the Asgard diversity that we see now was a hyperthermophilic organism that was able to fix CO2 via Wood-Lundell pathway, was able to potentially obtain acetate and utilize acetate, had a very clear fingerprint of organisms that live in extreme environments. However, this Asgard ancestor went through the evolutionary time and gave rise to the hot ancestor. This ancestor was the one who gave rise to complex life. And their metabolism is very different from the Asgard ancestor. We predicted that the ancestor that gave rise to complex life was heterotrophic, was not a hyperthermophilic organism, was mesophile, and had this specific complex gear, the electron transport change, that was able to respire potentially nitrate, and with this obtain more ATP. And this completely changes what we know about the history of how eukaryotes evolve, because before, as I show you in the figure, mitochondria was what we thought the reason why complex life originated. More ATP production of oxygen gave rise to complex life. But now we're predicting that potentially hot archaea were able to obtain ATP, which is completely fascinating. And now with our amazing PhD student, Katie, who's here in the audience, and her PhD, she is expanded this diversity of Asgard archaea. Now we have 1,000 of genomes of Asgard archaea. In the analysis that I showed before, we only had like 200. Now we have 1,000. And this is completely changing our understanding of Asgard diversity. We can look at the genomes, we can look at the evolutionary trajectories, we can look at the specific potential. The color ring that you see here is the potential that any given genome is able to be in, the likelihood of any given genome to be involved in the main cycles, if it's able to cycle nitrogen, carbon, oxygen, etc. And this is done with a computational algorithm that I developed during my PhD. And so with different techniques, we are able to predict whether different mechanisms in Asgard archaea. But now let's go back to the question. Are these archaea that we see today are able to produce the lipids that we see in the fossil record, right? So that's the question. And so far, there's no archaea ever known to be produce these lipids. So in order to tackle this, we not simply use bioinformatic approaches, right? So in order to tackle these questions, we need an integrative approach, as I told you before, micropaleomics. So here is the view of this approach. So we have bacteria that synthesize hopanoids. Bacteria now has been recently seen that also synthesize sterols. And we have eukaryotes that synthesize sterols. So now, in this approach, this is the methodology, the thought process of how we are searching for these lipids in Asgard archaea. We have to identify the gene or the protein that's able to synthesize these compounds in bacteria and eukaryotes. We do an homology search. We search in thousands of genomes and thousands of data, terabytes of data, more than 15 terabytes of data in order to search these genes. Then if we look at the genes, then we see which organism is the one that has these genes. We can then build evolutionary relationship of the specific protein. Then we can look at the genomic context and see, okay, this gene with the genome has these genes next to it and probably is involved in X, Y, and Z. We can also predict now with novel technologies, we can predict how that protein might look structurally. And then if we have good targets, we can clone them, meaning we can put them in E. coli, in a bacteria, that the bacteria is then going to produce those proteins. Then we can express them. We can see under certain conditions whether that protein is expressed, and then purify that protein. And once you have that purified, then you can see if the function of that protein is the same as the function that it's in the lipids that you find in the fossil record, right? So how do we start searching for them? In bacteria, all of the these lipids are triterpenoids, and as any isoprenoid, it's synthesized via these two precursors. I'm not going to go into the details. The only thing that you have to remember is bacteria use a different pathway, and archaea and eukaryotes use a different pathway to synthesize these precursors. And these precursors are the ones who synthesize isoprene units. And this is important just to kind of understanding why they are triterpenoids. So each isoprenoid unit, as you can see here, can fold and combine in different ways. And so we have monoterpene, which has 10 carbons and 2 isoprene units. Then we have sesquiterpenes, meaning 3 isoprene units, tetraterpenes. But the ones we're interested in is in the triterpenoids, sterols and hopanoids, which are 8 isoprenoid units and 40 carbons. And also I want you to remember the diterpenes, which are 4 isoprene units. And you will see why. So after several pathways, the important thing is that hopanoids and sterols are synthesized via squalene. So bacteria, via this specific enzyme, squalene hopane cyclase, can form hopanoids, and eukaryotes, via this enzyme called oxidosqualene cyclase, can form sterols. What's important about this is that even though we have different substrates, these 2 enzymes are evolutionary the same. They are evolutionary related, even though they are producing different compounds, they are coming from a common ancestor. So the question now is this enzyme, are these enzymes present in archaea? So we went ahead and searched in our data sets that we have built over this many years, that now we have more than 3,000 archaea representatives, 3.5. And all of these Asgard genomes. And if we look at by homology, we search computationally for these genes, I want you to focus on this last step. And here, I don't know, it doesn't look very good. Anyways, but we have different groups of archaea, of genomes of archaea, and we see that the size of the circle is how many sequences are found within each group. And surprisingly, there were a lot. But then when we look at the specific motifs, when we look at the specific fingerprints of the protein, we realize that only 7 among all of this display the conserved residues, meaning that the rest were noise. They were not true cyclases. So when we put these 7 sequences from Asgard in the tree, and this is done by our amazing collaborator, Hanan, from Stanford, a PhD student from Paula Velander's lab. When we look at the evolutionary relationships of the Asgard cyclases, we see something very strange that we were not expecting. But here we have the specific enzymes that synthesize sterols in bacteria and eukaryotes. And then we were like, okay, are any of this known in the fossil record? We are actually not sure. But what we saw is that they are close to the diterpenoid cyclases, which was very interesting. We were not expecting that. And then we went ahead and did a protein prediction. And the only information that you have to know is that by doing this type of analysis and looking at the active site of the enzyme, we realized that they have the specific amino acids to do the cyclation step, meaning that they can cyclase this squalenes or these isoprenoids. So everything was kind of okay. They potentially have something. They have the active sites, but they also have the active sites of things that look eukaryotic-like. So that gave us a lot of hope. And so then Hanan went ahead and did all of the molecular biology work to put these genes into a bacteria via heterologous expression to see whether or not they were producing these lipids. After a year and a half of analysis and bench work, and it wasn't really working because we were giving it a substrate that eukaryotes and bacteria uses, but not archaea. And so we finally, and this is a very preliminary data, we were able to obtain expression, meaning that they are actually producing diterpenoids. So experimentally testing and experimentally validating that they are producing diterpenoids, which is something that hasn't been seen in archaea, in any archaea before. So now we're at the step of purification, and once we are at the step of purification, everything so far indicates that they are diterpenoid cyclases. And what do we know about diterpenoids? They are not involved in the membrane, but this is what we know so far from information that's come mainly from plants. They are known as plant hormones. They regulate distinct different processes in plants, biological signaling, plant defense. They are also used as for cancer treatments, to treat malaria, but there is no information about the implication of these diterpenoids in the membrane, or even if they are found in the fossil record, because the methods to identify them are not meant to identify diterpenoids in the fossil record. So what I want to tackle is whether with microbiolomics, we were able to identify biological markers which are potentially diterpenoids. And so now the question will be if we can go to the fossil record and look for these diterpenoid compounds. And so another interesting thing is, what is the implication of finding these compounds in the Asgard archaea? Right now, we have lots of hypotheses. The archaea lipid divide, the lipid divide is to understand why eukaryotes share the same lipid composition as bacteria and not archaea, when everything is, as we saw, similar in archaea. So now what we're trying to understand is if we can really tackle what is the function of these diterpenoids, and potentially not involved in regulating the membrane as we know, but signaling some sort of process that gave us some sort of indication that there was some level of complexity. The pathway didn't evolve as it is. What we see now, it's going to be very different from what happened in the past. But now that we have all the way to the experimental testing of these molecules and actually work with that in the lab, we can start asking questions and going back to the fossil record. And by doing this, I think the take-home message is that collaboration between disciplines is super important, and it's essential to tackle one of the main questions in biology, which is the lipid divide. And so I hope that I can foster a collaboration and discussion after this talk. And with that, I want to thank, obviously, the Baker Lab and Brett, the amazing lab members, our funding sources, and our illustrators that have helped me do all of the lovely microbes that we see that I put on my presentation. And that with that, I will happy to take any questions. Happy to listen. Thank you.
I have a question about diterpenoid. You said like diterpenoid was never, like they never found it in archaea. It's just like because they didn't work on it, or it was just only on that specific type of archaea?
No, like in any archaea known so far. It hasn't been seen in any known archaea so far. Yeah, they have only been found in bacteria and eukaryotes. Yeah, so this will be the first time.
Gio has a question. It's he's asking, have you ever tried to express archaeal genes in eukaryotic systems? We're having a lot of trouble expressing genes in humans. And
If
that's the question. Yeah, I mean, he said that they're having trouble doing it essentially, but
yeah, to express genes in eukaryotic systems. So since we were having a lot of trouble expressing, and I think Hannah will have a very detailed answers for, but long story short was maybe the reason why it wasn't working when we were cloning these genes into E. coli was because potentially the system wasn't the right one. So we wanted to see whether we could clone them in Saccharomyces cerevisiae, which is an organism, a yeast, a eukaryotic organism, to try to express this. But after amazing troubleshooting of Hanon, was able to make it work in a bacterial system. But the other step was maybe try to do it in yeast and see if that works for you, Hale. Maybe that'll be the way to go. Any other questions?
Okay, let me ask the very naive response.
Yeah.
I found this completely fascinating, and it's an
Thank you.
introduction to a topic I know nothing about. But you started off with these lipid biomarkers that were found in the fossil record.
Yeah.
How do we know how true the molecules that we find today are to when they were involved in some living organism? Because there's been some tremendous time elapsed and they've been exposed to an environment.
Yeah, no, that's a very good question. So they have techniques. I don't know exactly. Jochen is an expert on this. Which you can actually see, extract them from the rock, any sediment or whatever, and ways in which you have to test whether they are as old as the rock, they are indigenous of the rock, like different metrics to see, okay, this is actually something that is as old as this rock itself, right? Now that that's checked, they have ways to extract this lipid from the record. There's an interview of Jochen and where he's talking about how he was extracting this lipids, and they were purple color. And it was very fascinating. So they have ways to see this. And then once you have the molecule in the lab, you can predict the specific structure, right? What is really fascinating about this lipid biomarkers is that, as I mentioned, they only lose some of the OH, and that's it. They don't change the structure as much, and that's why they are super important. The main structure remains the same, which is very fascinating. And yeah, I don't know if that answers your question. Yeah.
Yeah, I think I have somehow similar question. Like, how do you know it's not changing? But you need a reference. You have to say, okay, that is was original, but then you say it's not changing, but you need a reference.
Exactly.
Over millions of years, right? You have to understand the chemistry, what's going on in this, and how do you do that?
I have no idea how do you do that, but yeah. So you have the molecules of current organisms, right? As cholesterol. You have molecules such as specific molecules that you know the pattern. I don't know if I can draw something here, but you have the pattern in which this organism is being, measured, let's say it that way. And then you have the organism that you have that specific molecule produced by a living organism now, and then you compare both. And then there's ways, which I am not an expert on this, but there's ways in which you can compare a modern organism, such as bacteria, that are very easy to work in the lab, that produce this specific molecules, and then see if that molecules is similar to the fossil record. And what they have seen, right, is that the structure is the same. Only the OH or the hydroxyl groups are the ones that are lost. Yeah, but Paula and Jochen will have the specific answer for that. Yeah, it's very interesting. Yeah. Yeah.
So you look at the mass distribution and say, okay, level.
Yeah, but they have other ways to do it too. They have like more chemically detail. I don't know, Canon is here, but yeah.
Yeah, I mean, I'm
Yeah.
not, that is not our thing, so I'm.
Yeah, no, it's
It's witchcraft to me too.
yeah.
Will said, awesome work. Have your collaborators ever been able to, this is my question actually, able to examine the main lipids of Asgard archaea, other horticulture archaea? Do you have a sense of what the main lipids are in Asgard's?
Yeah, we're working on that. Yeah, we are characterizing this specific lipids in terms of their genomic information. We are doing the evolutionary trajectories of the genes that synthesize the lipids and the membrane. So we have the archaea, we have their membrane, and we follow all of the different routes that are necessary to synthesize the lipids that are here. So in that process, and which I didn't show a lot today because we're still working on that, and it's very preliminary, but there are key enzymes that are, again, thought to be unique to eukaryotes that we're seeing in Asgard. So once we have that solved, I will be able to talk about all of the synthesis of these lipids, and it's still very preliminary. But yeah, we're trying to do that. Yeah. Maybe.
Do you know if the Asgard archaea have ether-linked or ester-linked membranes yet, or that's still an open question?
Yeah. We'll are trying to figure it out. Yeah.
We don't know yet,
We don't know yet. We don't know yet.
but it's pretty damn fascinating what we're seeing.
Yeah.
I guess is what I would say.
Yeah.
We're trying to figure it out.
Yeah. Yeah, but so this cyclases, so our hypothesis was, okay, we have some of these molecules that were thought to be unique to eukaryotes and regulate key features in eukaryotes like cholesterol, and the membrane is key for this, right? And so we were very excited about the idea of having sterols in Asgard archaea, but that doesn't seem to be what happened. In reality, when we look at the evolutionary trajectories, it's way more complicated than we thought. When we look at other proteins within Asgard archaea, it's very clear that when you put them in a tree, you see Asgard and then eukaryotes. It's very clear that the Asgard archaea gave like our ancestors to those proteins. That's not what we're seeing, and it's way more complicated than we thought. But yeah, we're trying to get to that point. Yeah.
Okay, I think.
Yeah, is there any other question?
I think that covers it. There's lots of like anecdotal comments, but nothing we need to address. Okay, thanks.
Thank you.